Good day! Today we'll explore food microbiology and microbial biotechnology, how microorganisms influence the food we eat, from causing spoilage and disease to creating fermented foods and valuable industrial products. These tiny organisms are both our greatest enemies and most powerful allies in food science. Most foods provide ideal conditions for microbial growth. They're rich in organic matter, have enough moisture, and are usually neutral or slightly acidic. This makes them perfect ecosystems for bacteria, molds, and yeasts. But the mere presence of microorganisms doesn't always mean danger. Their uncontrolled growth can cause spoilage or even foodborne illness. However, under controlled conditions, microbes can actually improve food quality, as in fermentation. So, food microbiology focuses on understanding and controlling these organisms to ensure both food safety and desirable production. Different foods attract different spoilage microbes because of their composition. For instance, meat and fish spoil quickly because of surface contamination during handling. Eggs may develop black, green, or red rods depending on the bacteria involved. Bread can become soft and stringy, called ropy bread due to Bacillus species. Molds like Aspergillus flavus and grains and nuts produce aflatoxins, toxic compounds that cause liver damage. Spoilage changes the smell, taste, and color of food, making it unsafe or unappetizing. Understanding which microbes thrive on which foods helps us prevent contamination during processing and storage. Not all microbial activity is bad. In fact, fermentation is one of the oldest and most beneficial uses of microorganisms. Yeast like Saccharomyces cerevisiae ferment sugars to produce carbon dioxide and alcohol, the process that gives bread its soft, airy texture and beer its alcohol content. Molds such as Aspergillus oryzae help produce soy sauce by breaking down soy proteins and starches. In lactic fermentation, bacteria converts sugars into lactic acid, naturally preserving foods like pickles. These controlled microbial activities enhance flavor, extend shelf life, and improve nutrition, turning microbes from spoilage agents into preservation tools. Microbial biotechnology, also known as industrial microbiology, uses microorganisms to produce products of economic value. These range from enzymes used in food processing and detergents to antibiotics, biofertilizers, and even agents for waste degradation. Common enzyme-producing microbes include Aspergillus, Bacillus, and Pseudomonas. Enzymes like amylase break down starches, proteases digest proteins, and lipases target fats, all crucial in industries like food, leather, and pharmaceuticals. Modern molecular tools such as expression cloning and protein engineering are now used to enhance enzyme yields and stability, making industrial processes more efficient. In summary, microorganisms play a dual role in our lives. Some cause food spoilage and disease, while others contribute to food preservation, flavor development, and industrial production. Food microbiology teaches us how to limit the harmful effects and maximize the benefits. Meanwhile, microbial biotechnology pushes this further, turning microorganisms into tools for large-scale enzyme production, waste treatment, and more. By understanding and managing microbes, we transform natural risks into powerful opportunities.